based on the structures that are found in these electron microscope images. And so now, rather than a cartoon as we showed in that video, you see the actual neural circuits. And you can see in blue the presynaptic element, because it has vesicles which can release a neurotransmitter onto a postsynaptic spine-like structure. And so using this approach, Jeff Lichtman and colleagues at Harvard are able to reconstruct actual circuits in the brain. And this used to be a very, very painstaking process that would take years if you were going to reconstruct a cubic millimeter, a very, very small piece of brain. But in fact, he has automated almost every step of this process, including the cutting and the reconstruction, imaging and reconstruction. And in fact, you can label in color all of the axons, the inputs, onto this one dendrite of the receiving cell. And in fact, he likes to joke, it's amazing what you can get undergraduates to do for a letter of recommendation. They've colored in every single vesicle in these boutons. And this gives you some sense of how strong any one of these synapses might be. So the day has neighboring columns, other cells which might make a very specific inhibitory synapse here at the site where action potentials are generated. And in the ancient times when these were drawn, they looked like chandeliers. So each of the lamps were actually candles, and the candlestick holders of the chandelier were actually forming synapses here, and so on. And I guess they started to run out of names, and so it became more descriptive. It turns out transmitters are secreted at high amounts, and it's paired with the stimulus that trains the eye, which is weak. Another approach, very different, is exercise. So in fact, when mice run, it turns out that they secrete a lot of acetylcholine in their cerebral cortex. In this experiment, this mouse has been engineered to activate its locomotion or running center deep in the brain whenever a blue light comes on here, shining into that part of the brain. And so you'll see this animal is freely able to move, and when the light comes on, they start running. They don't know why they're running, but they start running. When the light turns off, they stop. And so in this condition, every time they run, you can pair the running with visual stimulation. And this is an adult animal. And what Stryker and colleagues found in this particular experiment is that pairing a visual pattern with running actually over several days leads to a strengthening of response to the stimulus that they were specifically watching while running, but not to any other stimuli that they were not seeing. And so this is a big sigh of relief that perhaps this reopening of plasticity is possible, but it's very specific to what the animal is experiencing at that time. If you look at where this LINX1 molecule is expressed, it turns out to be very interesting. It's expressed in a particular subtype of inhibitory neuron again, which has many of these neuromodulatory receptors, like the nicotinic receptor, serotonin receptor, and so on. And in this case, in the auditory cortex, if we in fact isolate these cells, and we look at the expression of LINX1, as I told you, it goes up with age. And the role is to dampen the response to nicotine. So in a young mouse, nicotine applied to these cells causes excitation. But as the animals get older, this excitation is dampened by LINX. If you remove the LINX1, even adult mice respond vigorously like a young animal. And so these cells turn out to be a new kind of inhibitory neuron that adds to this picture. So what I've told you today is that there is an excitatory-inhibitory balance which determines when these windows of plasticity happen. The actual change in the circuit is happening on these excitatory cells where spines can grow or be pruned. There are ever new players in this process, such as microglia. The effect of attention is coming most likely through inhibitory cells in the upper layers of cortex that are full of these receptors as well as LINX, and that provides context. When should plasticity happen? But at the end of the day, the convergence point seems to be these basket-type neurons here in the middle of the cortical layers. And as I told you at the beginning, they are eventually wrapped up in perineuronal nets, which trap these various excitatory and inhibitory synapses. And so we can now take this very broad concept of critical periods and turn it into a local circuit story. And with modern neuroscience techniques, we can actually start to dissect the circuitry underlying this power of the infant brain. I just want to end with an application of this because attention is something you can measure also outside the brain. There are many peripheral measures of heightened attention or arousal, and one that is well known is your pupil size, right? And if you look at the pupil of an individual who's aroused or attentive, it typically grows in size, and you can make the same measurements in mice. And in fact, you can measure spontaneous pupil fluctuations as an indication of the attention system. And in fact, the LINX1 knockout mouse gives us a great opportunity to train a deep neural network to detect any slight fluctuations in pupil size. So here's the setup. Just like you saw the mouse running on a wheel, you can measure the pupil size under low light conditions. And in a typical animal, they will fluctuate from small to large to small to large in this manner. And you can collect hundreds or thousands of these fluctuations and plot them from small to large size. In animals where the attention system is enhanced, you see that pupil fluctuations tend to be on the larger size. And in fact, you can train a machine learning network, an artificial neural network, to detect the difference between such animals at a very high accuracy. Once you've trained a network like this, you can test any mouse model you want, including a variety of models of neurodevelopmental disorders. And one example that we study heavily is called Rett syndrome. This is a mouse model of a disease in humans where one particular gene called MECP2 is defective. And in such animals, you also see that they have a heightened attentional or arousal states just by measuring the pupils. And quite satisfyingly, if you restore this gene only in the acetylcholine population, you can rescue completely this hyper-attentive, hyper-aroused state. And so it's very exciting because it suggests we can measure from outside the brain what the conditions are inside the head. And you can train a neural network which will accurately predict, in fact, whether the animal is at a particular stage of its trajectory. And that's all well and good, but who cares about mice, right? We want to care about humans. And so the beauty of the deep neural network is that you can retrain the network in a process called transfer learning on data from humans. And in this case, we used available data of attention or arousal from heart rate. Heart rate variability, like pupil variability, is a proxy of arousal. We can gather data from the clinic at Boston Children's, retrain a network that was initially trained on the mouse, and accurately predict which kids will go on to regress in this process. So it's a very exciting time in neuroscience, especially developmental neuroscience, because these age-old concepts that the early brain is plastic and goes through critical periods can be understood at some deep biological level. And by doing that, we can get some insight into derailed development, like neurodevelopmental disorders. But it also gives us this science fiction-like possibility of reopening a critical period later in life. And you can imagine how important this could be if you suffer a stroke or some kind of traumatic brain injury in adulthood. Wouldn't it be great if you could make your brain young again and try to rewire the non-damaged parts of your brain to recover some of the function that was lost? And in fact, that day has really come upon us, and it's a very important issue, not only for scientists, but also for social scientists, because you can imagine taking this kind of finding and going too far, as Hollywood often likes to do. I will leave you on this thought that critical periods most likely have evolved for a good reason, and excitingly we can discover why that has happened. But we also need to be mindful of why they close. That perhaps balancing brain plasticity is just as important as the initial rewiring that's allowed to set up our brains in the first place. I'm happy to take any questions if you want to stick around. A stimulus appears and get a reward in the back of the cage. And so this mouse has learned to do this task. But you can make it attentionally demanding by having the visual stimulus appear for a very brief time. So in that case, the animal wasn't paying attention and missed the stimulus. There's no reward, and a light comes up. That's a pretty typical mouse. If you remove this Lynx 1 brake on attention, this is how the animals behave. You can see immediately from their body language, they are very attentive. This is the short stimulus. It gets it right away. And trial after trial, these animals are right up at the front of the screen, on task, doing their job. They make very few mistakes. And I think they're some of the best students in my lab. So the prediction is these animals have a childlike, heightened attention to stimuli. What happens to their critical period? Well, if we go back to the visual system model, you can measure vision from these animals, and you find that their vision is typical of mice, around 0.5 cycles per degree. If you patch one eye, this process here in black, during the typical critical period, these animals with no Lynx 1 will also lose vision. But unlike a typical wild-type animal, where reopening the eye after the critical period has no further benefit, so the vision stays poor, that's why it's called the critical period, these animals without Lynx can fully recover vision. And so it suggests that their critical period stays open longer. And in fact, it makes this molecule a very exciting target for drug discovery to make something that can make our brain young again. But we already know how it works. It's dampening the nicotinic receptor. So one idea is to maybe prevent this break from having its full effect. And there are drugs that prevent the breakdown of acetylcholine. And so this neurotransmitter can last a little bit longer than normal. And maybe that alone is enough to override this break. So if you give a wild-type animal this drug, acetylcholinesterase inhibitor, then in fact you can rescue their vision as well. And as I told you at the beginning, this condition of amblyopia has no cure currently, and so this is quite exciting. We can at least cure vision in mice, and so it's led to a clinical trial which is ongoing at Boston Children's Hospital, because these drugs are already clinically available for other conditions where acetylcholine is weakened. And we are now testing whether we can rescue vision after the critical period by reopening brain plasticity.